Hello everyone, welcome to Unit 4 Photosynthesis lecture video number 6, where we're going to begin to go over the two stages of photosynthesis starting today with the light reactions. I will have to talk fairly quick since there is a large amount of information to cover during this video. Please feel free to pause it and rewind it at any point. So as I said, there's going to be two steps to photosynthesis, and those are going to be what we call the light reactions as well as the Calvin cycle. All of this will be taking place here within a chloroplast. The light reactions, their goal will be to produce ATP and NADPH using light, and then that chemical energy will be used in the second stage of photosynthesis called the Calvin cycle to eventually make sugar or glucose. So as you see in this diagram for the chloroplast, light and water will be used in the thylakoids to produce ATP and NADPH, and oxygen will be released as a waste product. The energy from the light reactions will then later be used in the Calvin cycle to turn CO2 from the atmosphere into sugar. Alright, for step 1, the light-dependent reactions, which will be the focus of this lecture. The goal is to make ATP, as I just said, and NADPH. These will be used as the energy source to turn carbon dioxide in the Calvin cycle into glucose. This process is going to take place right here in the thylakoids. So that's every one of those individual discs. And again, a stack of thylakoids is called a granome, is where the light reactions are taking place. We need several things for this to occur. Our inputs are light energy, water, which will be split and produce oxygen as a waste product. Oxygen gas will diffuse out of the thylakoids into the chloroplast, into the cell, into the leaf, and will eventually exit through the stoma, the openings between the guard cells on a leaf. Not the stroma, the stoma. We will also need ADP and a third phosphate group so we can regenerate them into ATP. And NADP plus, an electron carrier, will be charged up and reduced to produce NADPH, which we said both ATP and NADPH will be used later on already in the Calvin cycle. So in this animation you see happening, all right, it may look a little intimidating at first, but as I go through the steps, I want you to break down the light reactions, which is what you see here, into two categories: electron movement and hydrogen ion movement. All right, so our focus is here along a thylakoid membrane of just one of the thylakoids. This is what we're seeing in. The green blobs would be photosystems that we previously discussed. Blue, yellow, and orange would be integral and peripheral proteins making up what we call an electron transport chain. That is transferring electrons from PS2 to PS1. And then we have ATP synthase that we've discussed previously that is going to use this large hydrogen ion gradient to make ATP. So electron flow is going to occur between photosystems to make NADPH. And as the electrons flow, you create a hydrogen ion gradient in the lumen, which is used by ATP synthase to spin like a turbine to make ATP. So here's those two products up above, NADPH and ATP, that are being made in the light reactions. So let's dive into how this is going on. So I have a diagram here that's already numbered. I'm going to use these numbers that you see in the diagram, all right, 1 through 6, all right, to go through the steps of photosynthesis. So here we are on a thylakoid membrane. In this diagram, we can see PS stands for photosystem. I can see one photosystem here and another over here. In between them, I have an electron transport chain, which is what we saw in the previous animation. Blue, yellow, and orangey red blobs moving. And then over here will be a shorter electron transport chain that will eventually be used to make NADPH. So let's begin. Step 1 is light strikes photosystem 2, causing all of its pigments electrons to become excited. So we can see the flashing yellow symbol for light. It's striking photosystem 2 here, exciting all these electrons, and eventually it's going to cause this last pigment molecule, which is chlorophyll A, sometimes also referred to as P680, to lose a pair of electrons. All right, when it gets excited from all the light energy being transferred through it, it literally loses a pair of electrons, and here they are. They get thrown up to a higher energy state. Those electrons are eventually going to be fed into this electron transport chain. But before we get there, let's focus on replacing these two electrons that were just lost. And that's going to happen here in step 3. In step 3, water is going to be split. There's a technical term for that. It's called photolysis. Photo means light, lysis means split. So light energy is going to be used to split a molecule of water. When water is split, you immediately feed 2 electrons right back in to PS2. So right here, these will replace the 2 electrons that were just lost. But we're also going to get 2 other products that are going to start to build up here in the lumen, the inside of the thylakoid. And those are going to be hydrogen ions and oxygen. Here it says half of O. All right, what that means is it's not an O2 molecule. All right, I need 2 oxygens to make 1 breathable O2 molecule. So this is just one of them. So from 1 water, I only get 1 oxygen. So you'd have to split 2 water molecules in order to actually produce 1 breathable O2 molecule. But it's still oxygen. So when we split water from photolysis, 2 electrons are fed back into PS2. And in the lumen, we start to get hydrogen ions as well as oxygen being produced. Our next step is now to follow these 2 electrons that PS2 loses down the electron transport chain. So for step 4, the electrons lost from PS2 here are going to move down this electron transport chain towards PS1. All right, the 2 electrons right here from PS2, as they move down the ETC, they're going to pull additional hydrogen ions from the stroma down into the lumen, the inside of the thylakoid. So those 2 electrons you see flashing right there, being lost by PS2, will move down the electron transport chain. And as they do, since they're negatively charged, they'll attract more hydrogen ions that are in the stroma and suck them against their gradient down into the lumen. So I'm producing an extremely high concentration of hydrogen ions here in the lumen from both the splitting of water molecules as well as the movement of electrons from PS2 to PS1. You have additional ones being pulled from the stroma down back into the lumen. Now let's focus on step 5 over here. Step 5 is going to be at the same time that PS2 is losing a pair of electrons when it gets hit by light. Again, beams of light are also called photons. PS1 is also getting hit at the same time. It loses a pair of electrons right here that are flashing, the same as PS2 did. However, I don't see water here, do you? Water isn't being split. Water is not being split to replace these 2 electrons that PS1 is losing. So they are going to get replaced by these 2 electrons that are actually coming from PS2. So 2 electrons lost by PS2 are replaced by splitting water. And as these 2 electrons from PS2 move down the ETC, they will replace a pair of electrons that are also getting lost by PS1. And then finally, for step 6, all right, NADP plus you see flashing is going to be the final electron acceptor. Eventually, the flow of electrons has to cease. So NADP plus will pick up the 2 electrons that just came from PS1. When it picks them up, it becomes reduced. And your final charged electron carrier is NADPH. And this is going to be used in the stroma the same side of the membrane that it was just produced on, to produce glucose. So what we just saw was the flow of electrons, starting with water, replacing 2 electrons that PS2 loses. Those 2 electrons from PS2 move down the ETC, replace the 2 electrons that PS1 loses, which are then given to NADP+, reducing it into the fully charged molecule that we call NADPH. So that's your full electron flow in the light reactions. All right, I think it's easier to think of the flow first and how it's used to make NADPH. Now for the last portion, we can focus on this high hydrogen ion concentration that we built again one of 2 ways: splitting water from photolysis and pulling in additional hydrogen ions from the stroma into the lumen as these electrons move down the ETC between photosystems. So that's our last focus now, is how we're going to use those to make ATP. We're going to do that in step 7 in a process called photophosphorylation. Let me interpret. Photo means light. Phosphorylate means to add a phosphate group. So light energy has been used to create this hydrogen ion gradient you see down below here in the lumen. Remember, as electrons move from photosystem 2 down to the ETC to photosystem 1, we eventually make NADPH here. But we also created this hydrogen ion gradient, one more time, from splitting water over here at PS2 and pulling hydrogen ions from the stroma into the lumen as they flow down the ETC. So now what's going to happen is these hydrogen ions are going to flow naturally from high to low, so from the lumen back out to the stroma through the enzyme we have already learned about at the very beginning of our unit, ATP synthase, which will spin like a turbine to regenerate ATP. So that's what photophosphorylation means. So the hydrogen ions will flow from the lumen, so this gradient we produce here, back out to the stroma through ATP synthase, which helps to drive the production of ATP. So you're phosphorylating ADP, adding a third phosphate group onto it to make ATP once more. So if I draw your attention here in the diagram, you're going to see a flashing hydrogen. That represents the large gradient that's inside the lumen. That's going to flow naturally from high to low through ATP synthase, back out to the stroma. And as it does, that provides the kinetic rotational energy, is what we described earlier, to help spin ATP synthase. As it spins, think of it as clicking 1 second at a time on a clock. Every time it clicks around once, that provides the momentum needed to add a phosphate group back on the ADP to make ATP. So now, at the end of the light reactions, electron flow has produced NADPH, hydrogen ion flow has been used to create ATP, and these 2 molecules here that were just made in the stroma, again, these 2 blue dots here will be the electrons that have flown through, will be used by the Calvin cycle to turn carbon dioxide into glucose or sugar. So again, you can see here in conclusion, we're making NADPH and ATP in order to make glucose later on during the Calvin cycle. And here in this animation, this represents all of the photosystems and electron transport chains. Here is ATP synthase. 2 electrons are lost by PS2, travel down this ETC, replace 2 electrons lost here by PS1, which are used to make, right here at this point, NADPH. You'll see this blue molecule come in. Now you have your hydrogen ion gradient made by splitting water right there, as well as the electrons move down this ETC right here, they're going to pull more hydrogen ions into the lumen. And in conclusion, the process of photophosphorylation will be now using this hydrogen ion gradient to flow through ATP synthase, spin it like a turbine, as you can see, to rephosphorylate ADP to create ATP right here.